Today we're going to be looking at the biggest PowerPoint that you've ever seen on meiosis. Let's begin. So meiosis is in Pearson Biology 12 on section 6.3, page 312 onwards. So the syllabus objectives for this lesson are: within the process of meiosis 1 and 2, recognize the role of homologous chromosomes, describe the processes of crossing over and recombination, and demonstrate how they contribute to genetic variation. Now, a note that I want to start with, which is really important. The syllabus, we used to talk a lot about mitosis. The syllabus doesn't require us to talk about that anymore. But in my years of teaching biology, I really think that the best way to understand meiosis is to go back over mitosis and the cell cycle. You can skip through these, and I've labeled them as background information, but I think that the best way to proceed, especially if you look at meiosis and you're a bit confused about what's going on, is to go back to mitosis and look at the basics again. Just be mindful that it's not a requirement for you to know and rattle off all of the stages of mitosis, but it is easier for your understanding going forward. So we're going to start with mitosis, and as you can see, all of these upcoming slides are labeled as background information. So feel free to skip ahead if you're feeling confident with the content, but if you do feel you need that extra background, I'd be starting here. It's also important to note that Pearson 12 also spend a whole section talking about mitosis. So even though it's not a syllabus dot point, they've obviously still valued it as an important topic to know. Now, we would have talked about mitosis when you guys did cell division way back in year 8 and year 10. Mitosis is the cell division that happens when our cells need to make new cells. So it's happening when we're growing, when we are replenishing damaged cells and things like that. And when mitosis happens, the daughter cells are genetically identical to the parent cells. We're not doing any mixing of DNA or adding in any new DNA or variation to that. We're simply copying and pasting the original cell. I spoke about the cell cycle in the previous PowerPoint looking at DNA replication, and we will go into this a little bit more in this lesson. So just to recap, with our eukaryotic cells, the cell division processes undergo this cell cycle. This is kind of what tells you the stage of where the cell is at in terms of its division. Now, in the last lesson, we talked about this yellow phase, which is interphase. And in that phase, that's where we're really looking at the cell growing, copying its contents, and getting ready for division. Mitosis is this green section, which is the actual dividing part. And cytokinesis is this last part here in the blue, where we have the cell actually splitting into two. This is another representation of that. And I like this one because you can clearly see what's happening at each stage. And we'll talk about this G0 phase as well. Now, some cells are dividing a lot more rapidly than others. So things like embryos, when they're developing, the skin cells and your gut cells in your stomach lining, they're dividing all the time because they're either growing really quickly or they need to be replenished because they get damaged. So the cell cycle is continuing constantly for those cells. Other cells don't continually divide. So things like your nerve cells, they typically aren't known for being able to replenish themselves or make new copies of themselves. And that's what we're going to look at when we look at this G0 cycle, where they pretty much do the cycle and then leave the cycle and stop undergoing any further divisions. So our 4 phases of the cell cycle. We start with this phase here, which is G1. It's also called gap 1. And in that phase, all of the contents of the cell, so all the organelles, everything except for the DNA, get copied. So at one point in time, you've got double the mitochondria, double of everything, and everything is getting ready in the cell to split into 2. The S phase here is short for synthesis, and this is where the chromosomes are duplicated. That is what we went through in the last PowerPoint in our DNA replication phase. DNA replication happens in S phase. After S phase, we've got the G2 phase, which is also called gap2, which is a double checking sort of phase. All it is your DNA, checking mechanisms happen, and you've got little helper enzymes and things moving along the newly replicated DNA to make sure that there's no damage and no repair that needs to be done prior to the cell dividing. Now again, remember that this G1, S and G2, that entire part is labeled as interphase, which you can see on this inner little circle here. Then we have M, or mitosis phase. So when you look at this in terms of a cell's life cycle, mitosis, or the actual splitting part, is only a very small piece of the puzzle. Majority of the time for a cell is spent in copying its contents, copying its DNA, and checking. Now, when this becomes out of control, that's what happens with cancerous cells. Rather than spending all of this time in G1, S, and G2, the cell is spending way too much time doing mitosis, and it's dividing way more rapidly than it should, and it's out of control. Now, cytokinesis here is that last step where we finally get two cells budding off. And this G0 phase here is really important because, like I said, with particular cells like nerve cells, once they've undergone division and we've got our newly formed cells, those cells leave the cell cycle, and they call it cell cycle arrest. And that means that those cells are no longer going to be undergoing any type of cell division. And cell division is complete for those cell types. Now, in your PowerPoints, there's this link to the phases of the cell cycle that you can go through if you like, but again, remember, it's background information. Now, these slides are just going over again what's happening in those phases. So really quickly, gap 1 is happening between our mitosis and DNA synthesis. We've only got one singular chromosome existing as one chromatid. The cell grows and performs its normal functions in that phase. In the S phase, DNA is replicated, the chromosomes double, and so now we have full chromosomes rather than singular chromatids. The gap 2 phase is that checking phase where the cells are getting ready for division, last little bits of finalization. And remember that those three phases, G1, S, and G2, are grouped as interphase. So before we start looking at the processes of mitosis and the phases, it's important to understand the different structures that we're going to be looking at. And the chromosome is this typical X-shaped structure here, but a chromosome is made up of two separate chromatids. So these two halves, like the left and right parts, are called chromatids. And so we say that these two are sister chromatids because they're a copy of each other. The other important thing is the centromere, which is this middle region that holds the two chromatids together. We're going to have a look at this shortly, but this is where the mitotic machinery that we're going to look at grab onto to pull apart the two chromatids and split the cell and make one chromatid go to each cell. All right, so you can see our chromosomes lined up here, and each one of these has their sister chromatids, and you can see that there is the centromere part of the chromosome there. Now, in terms of the mitotic machinery inside the cell, we haven't often talked about these organelles called centrioles, and they exist in the cell, but their sole job is to assist with cell division. The centrioles make up this component of the mitotic machinery, which we call the centrosome. And the centrosome is basically the centrioles as well as these microtubules that extend out from the centrosome, which form what we call a spindle. The microtubules extend out and attach themselves to the centromere so that when the spindle contracts, it's going to pull apart the two chromatids and take one to each side. The whole structure is called a spindle, and the spindle is made up of these microtubule fibers that extend from the centrosome. So this is a better picture to show you what this looks like of the centrosome. Remember, you have one of these at either end of the cell. Inside here, you've got your centrioles, these two sort of cylindrical organelles. Don't worry too much about them. The important thing is that you know that you have microtubules, which are going to be progressing out of your centrosome, and they are forming the spindles which go along and grab onto the chromosomes during cell division. Now again, we've got our centrosome at each end with our centriole and our microtubules forming a spindle. So these microtubules are attached to the centrosome at one end, and they are attached to this part of the chromosome called the centromere, which we'll have a look at shortly. And remember, in that metaphase part, all of the chromosomes line up along the middle of the cell, and it's these spindles that come and latch on to pull apart our chromosomes. Now, as I've said, you don't need to know all of the stages of mitosis. However, you need to know the stages of meiosis. So it's often a lot easier if you already understand this, so that when we proceed with meiosis, it makes a lot more sense. Now, when our cells are doing mitosis, remember, they're just copying and pasting the information. We're not mixing any genes or adding any variation into here. It's just a straightforward process. Now, the phases of mitosis, and these are the same for meiosis, are prophase, metaphase, anaphase, telophase, and then our cytokinesis happens at the end when we get our daughter cells. The following slides are going to go through each of these, but as a quick summary, in prophase, you can see that the nucleus here is starting to break down. You've got these little gaps in the nucleus, which suggests that the nuclear membrane is degrading. You've got the spindle starting to form at the end here, and we'll talk about that soon. In metaphase, your chromosomes have now left the nucleus and they're lining up in the middle of the cell, which we call the equator. It's important to remember too that we actually have 23 pairs of chromosomes here. We don't have 4, but for the ease of showing this, we simplify it. Now they align at the equator and these spindle fibers form, and they're going to come along and grab onto our chromosomes that are at the middle. In anaphase, we get the spindle fibers contracting, and what happens is now that they've attached to our chromosomes, when they contract, they're going to pull apart our chromosomes and pull a separate chromatid to each side of the cell. In telophase, they're really stretched all the way to opposite ends, and we start to see this budding happening, and we start to see the nuclear pore starting to reassemble back around our chromatids. In cytokinesis, we've got 2 newly formed cells, and they're going to go back to their original state, and again they can come back around and start this process again when they're ready for their next round of division. There is a video that you can watch on this to summarize those phases. So we will go through these in a little bit more detail now so that you can see what's happening here. So let's have a look at this in a little bit more detail now. Remember, interphase was the gap 1, S, and gap 2 phases of the cell cycle. It's the longest part, and it's where everything is copying and getting ready for cell division. As you can see in the picture, it just looks like a normal cell. There's no sort of formation of the spindle yet. The centrioles haven't moved to the 2 sides of the cell. It's just a normal cell, but it's copying everything in preparation. So in prophase, I mentioned this earlier, that we start to see the nuclear envelope degrade. So that's your actual membrane of your nucleus. And this needs to degrade so that the chromosomes can leave and align themselves in the cell. The chromosomes also condense, so they form these long, elongated, typical chromosome structures, which makes them easier to pull apart. We start to see our centrioles, which form our centrosomes, starting to move to opposite ends of the cell. So they would have been chilling up here before, and they've started to move to the opposite sides to prepare for mitosis. We start to see the spindle forming. So you can see microtubules starting to assemble and project out from the centrosomes. In metaphase, you've got the nuclear membrane completely gone, and you've got these condensed chromosomes aligning at the equator of the cell. Sometimes you'll hear this called the metaphase plate. We can see our centrioles have moved to the two ends of the cell, and they have projected microtubules out to form the spindle. They've grabbed onto our chromosomes at the centromere here, and they are ready to contract and begin the next phase. All right, in anaphase, we now have contracting of our spindle fibers, and they are going to latch on to either side of the chromosome, and as they contract, pull one chromatid from the chromosome to each side of the newly forming cells. In telophase, we're really close to forming two new cells. We can see the nuclear membrane starting to reform around our new copied chromosomes. We can see the spindle starting to disintegrate and go back to its original state. And then finally, cytokinesis is that last step where the two cells break off and we have newly forming cytoplasm and two new daughter cells. As I said, it's really helpful if you understand those stages of mitosis, because they're the basis for what's going to be happening in meiosis. Now, meiosis is a different type of cell division. I mentioned mitosis being important for the division of our body cells, like skin cells and gut cells. Meiosis is important as it is the cell division that happens in order to make our sex cells. So this is how our chromosomes that we inherited from our parents are going to end up in the egg and sperm cells to pass on and be inherited by our offspring. All right, so those body cells that I mentioned before, like the skin cells, we call those somatic cells, and they're going to be undergoing normal mitosis because it's just a copying and pasting of our normal cells. With our meiosis, that's happening in the germ cells. And germ cells are the cells that are going to give rise to sex cells or gametes, such as egg and sperm. So remember, with sexual reproduction, we've got the joining of male and female sex cells, which are gametes. And that's going to involve the egg or the ovum and the sperm. In order for the egg and sperm to actually get made, we have germ cells. And we're going to look at how those germ cells turn into mature egg and sperm cells. Now, this is really important because this is the process of meiosis. And this is much different to mitosis in that each one of our gametes is unique from one another. Unlike mitosis, we're not making copy and paste versions of our cells. Each egg cell in a woman has a different combination of chromosomes and a different genetic makeup. With mitosis, every cell is identical. So that's the key difference that we want to be looking at as we proceed forward. Now, meiosis is specific to the sex cells. It's the only time where meiosis occurs, and it happens in the gonads. So for the female, that's going to be in the ovary, and for the male, that's going to be in the testes. Over here on the right, you can see we've got a sperm cell here. We call these haploid because sex cells have half as many chromosomes as what our body cells have. So rather than having 23 pairs like we have in all of our body cells, haploid cells only have 23 singular chromosomes. The same with the egg down here. It's haploid. It has 23 singular chromosomes. So not only is meiosis different because we are allowing variation among our different daughter cells, we're also halving the number of chromosomes. We're going from 46 chromosomes in our normal body cells into 23 for the sex cells. And that's important because our 23 from the male and 23 from the female need to join to make a diploid fertilized egg. Diploid meaning the full set of 46 chromosomes. Remember, a zygote is the name for a fertilized egg. So again, the purpose of meiosis is to produce gametes. That's our sex cells. The nucleus within our somatic cells, remember our body cells, have 46 chromosomes. We call them diploid, di meaning we have 2 copies, one from each parent, and you also see this denoted as 2N. The nucleus of our sex cells only contain 23 chromosomes, so we call them haploid, and they're represented as N. So rather than having 2 of each copy, they are a singular set of chromosomes. Therefore, with meiosis, the daughter cells only have half as many chromosomes as the body cells. And during fertilization, 2 haploid sex cells are going to join to make a diploid zygote. It's really important that you understand that terminology to assist going forward with this lesson. So just to recap, meiotic cell divisions in males and females, their specific job is to produce haploid sex cells. That is so that when fertilization happens, 2 haploid cells can unite and form a diploid fertilized egg. The fertilized egg can then undergo normal mitotic divisions to develop into a baby. On the right, you can see this represented in the image from Pearson. We have a woman here who has 46 chromosomes in her body cells, 2N or diploid, and same with the male. In the gonads, so the ovary and the testes, we have meiosis taking place, and we'll talk about how this is different for males and females in the next PowerPoint. Meiosis is going to take place to turn those 2N body cells into N haploid sex cells, so the egg and the sperm. And when they unite during fertilization, we have a 2N zygote, and then mitosis can occur. So we've now introduced the DNA, it's complete, and the fertilized egg has a full set of genetic information, and now that zygote can divide by normal mitosis to develop into a child. It's also important to recognize that chromosomes exist in different states depending on which part of the cell cycle we're looking at. So before DNA replication, so during interphase, the chromosomes look like this, which isn't what we're used to seeing chromosomes look like, and they're just existing as singular chromatids. There's still a centromere, but it's just holding the 2 ends together. It's no longer got that X structure about it. When the DNA replicates, so during that S phase of interphase, we're going to copy all of that. So we're going to have 2 chromatids now instead of 1, and they're just a copy and paste of each other because it's just mitosis. So you can see over here our 2 sister chromatids joined together at the centromere. But another important piece of terminology to know is that we get 1 copy of each chromosome from each parent. So this might be the chromosome that we inherited from our mum, and this might be from our dad, and they code for the same genetic information. So we call those homologous chromosomes. They're the chromosome pair that we've inherited from each parent that codes the same information. Now this bit tends to confuse people a little bit. And if we think about meiosis taking place in a male, for example, the sperm cells are being produced all through adulthood. So meiosis is occurring in the testes all the time. Now, when that happens, the man's genetic information has come from his mother and his father. So we have to think about their genetic material being mixed and combined to make his sex cells. So if we have a look at this, we have the chromosome here from the father's side, the paternal side, and the chromosome here from the mother's side. Now, when meiosis is happening in the sex cells, the chromosome pairs, the homologous chromosomes, come together and form what we call a tetrad. And this is basically two homologous chromosomes coming together. And then what happens is there's a shuffling and mixing of the DNA between these two chromosomes. We'll have a look at this in more detail soon, but this is really important in understanding why each individual sperm cell would have a different combination of DNA. And this is just showing those different states of chromosomes again. So remember that before DNA is replicated, we have one singular chromatid. It duplicates and forms a full chromosome containing two sister chromatids. And then during mitosis, that can then split again into singular chromatids. Now, to orientate you with the phases of meiosis, the phases are named the same as mitosis, and there are a lot of similarities with just some key differences. We can group these into meiosis 1, which you can see here, and there is one division event happening there. And then following that, we have meiosis 2, where we have a second division event occurring. In meiosis 1, we have those homologous chromosome pairs, one which we got from our mother and one that we got from our father. And in meiosis 1, those homologous pairs have to separate and move to different sides of the cell. And we have this recombining of genetic information happening there. So the first division step just separates those homologous pairs. The second division event, meiosis 2, is much like mitosis, where we have the two sister chromatids getting pulled apart, and we end up with haploid daughter cells that only have singular chromatids rather than chromosomes. So let's start with meiosis 1, having a look at interphase. So remember, before entering meiosis, the cell has to undergo interphase, just like we saw with mitosis. And it's the same where it has gap 1, S, and gap 2 stages. Our chromosomes have duplicated, so we have our full set of 46 chromosomes, half from our mum, half from our dad, existing in these full duplicated states. So just like mitosis, we start with prophase, but we call it prophase 1 as it's part of meiosis 1. During this phase, the homologous chromosome pairs, the ones that we inherited from each parent, condense together like I showed in this figure, and they form a tetrad. You can see this shown in much like what we saw with mitosis down the bottom here, where our two homologous chromosomes come together. And there's a really important phase that occurs where the DNA is crossed over. We call it crossing over. And see how this blue chromosome now has a little piece of green, and the green chromosome has a little piece of blue. There's actually a lot more crossovers than that, but it's just for ease of showing this. The same thing happens. We see the nuclear membrane break down. We see the spindle starting to move into its position. The real key difference here, though, is that we have a swapping of genetic information, and this happens randomly, so that the two cells that are going to form it from the end of this are going to be unique and have different mixtures of genetic information. This is that crossing over phase. You can see here we've got homologous chromosomes. They come together and form a tetrad, and they do this exchanging of material. And so now we have two unique pieces of DNA or chromosomes that are different from one another. And this is how we get our variation in each of the sex cells. This is why we're not all identical, because all of the sex cells will have a different combination of this DNA. Remember too that this isn't just happening in one chromosome pair, there's 23 pairs. So each sex cell is going to have a completely unique setup of the genetic information. We call this recombination or crossing over. Showing this again in a different way, homologous chromosomes form the tetrad, crossing over, and we have recombination occurring. This is showing this again, where you have one chromosome at the back, one at the front. They come together and cross over that genetic information. Sometimes you'll see them called homologs rather than homologous chromosomes. And the fancy name for the structure that forms to assist the crossing over is called a synaptonemal complex. Now, you might also come across this term, which are called chiasmata. And chiasmata are the crossover points, and we can observe these microscopically. Here you can see two homologous chromosomes. There's the centromeres holding the chromosomes together, and you can see where they've crossed over at these points. So they're called chiasmata, and the plural term for that is chiasma. You can see that again here in this more schematic diagram. So in terms of crossing over, it's common for multiple crossovers to happen. And when I search this up, it says around 25, up to 25 for each pair. So when you see those diagrams with the chiasmata, often there's a lot more than what is shown in the diagrams. Important point when we talk about the random nature of which gametes end up with what genetic material, the crossover points are random. So it's always going to be a random chance what DNA or pieces of those chromosomes end up in which gamete. So there's always going to be unique combinations, and a lot of variety can happen from that. All right, so moving on to metaphase of meiosis 1. So just like metaphase of mitosis, we have the spindle forming here. You've got the centrioles at either side of the cell, microtubules extended. But see how the difference here is that we have homologous chromosome pairs rather than one singular copy of the chromosomes. So the purpose of meiosis 1 is separating the two homologous chromosomes. Crossing over has happened. So you can see where we're going to pull this green one to this side, and we're going to take the blue one to this side, and that's the first division event of meiosis. In the second event, we pull apart the two sister chromatids. So in this part, we're going to have the microtubule attaching to the entire chromosome here at the centromere, and then as we'll see in anaphase, that's going to contract and move the chromosome in its entirety to each side. So you can see here, you've still got your whole chromosome, and it's being pulled to the respective side of the cell, and you can see that the recombination event has taken place, the crossing over. And remember that the crossover points would be a lot more complex than just this one little difference shown in the picture. Okay, telophase 1, just like mitosis, the chromosomes are at either end of the cell and they start to bud off from one another to produce two daughter cells. And then we prepare for the second round of division in meiosis 2. All right, so now that we've separated the homologous chromosomes from each other, we now move on to the second meiotic event, which is meiosis 2, where we're going to separate the two sister chromatids. So this is very much like mitosis because we're just dividing two chromatids of a chromosome to different sides of the cell. The key difference here is that at the end, we're going to have daughter cells which are haploid. So we're not going to be, we haven't duplicated the DNA and then split them. We're now splitting them so that the daughter cells are N and not 2N. So again, we have prophase 2. So just like in mitosis, the chromosomes are going to condense and the centrosomes are going to move apart and start to orientate themselves on either side of the cell. But notice how the difference here is that the chromosomes are shown having already crossed over. So we've got to keep in mind that the key difference is that these cells are not the same. This cell has a different genetic makeup to this cell, and they're going to divide again to have 4 daughter cells that are all unique, and they're going from diploid to haploid. Metaphase 2, so we have the microtubules aligned at the ends of the cells. We have our chromosomes lined up at the cell equator, and we have the spindle coming out and attaching on to those centromeres of our chromosomes. Now we're going to be pulling apart the chromatids, whereas in meiosis 1, remember, you had 2 homologous pairs. So we actually were separating the chromosomes. Now we're just separating the chromatids. Anaphase 2, we're pulling apart those chromatids to either side of the cell, and you can see how we're going to end up with a variety of different genetic material going to each of the sides of the cell. And telophase 2, the nuclear membranes reform around our new daughter cells. We have cytokinesis occurring to split the new cells apart, and the end product is we're going to have 4 haploid sex cells. So we now have an N set of chromosomes, and that will be what happens in our egg and our sperm cells so that they can join and make a diploid organism. This is a comparison to give you just get your head around the 2 different types of division. So remember, in mitosis, you've got your 4 phases plus interphase. It happens only in body cells or somatic cells. The purpose is for cellular proliferation, so that's growth and making new cells. We get 2 diploid daughter cells. The chromosome number stays the same, so it's still 2N. And we don't get any genetic variation. We're making a copy and paste of the same cell. In meiosis, we have 8 stages because there's 2 rounds of division. It only happens in the germ cells, the cells that are going to become the egg and sperm. The purpose is for sexual reproduction, so that we have gametes that have half the chromosome number in order for fertilization to occur. We end up with 4 haploid daughter cells rather than 2 diploid cells. The chromosome number is halved, and we have genetic variation as a result of that crossing over. Over to the right here, you can't see because I've hidden it with my big old face, but you've got mitosis going from 46 chromosomes to 46 chromosomes, and it's still diploid. Meiosis has 46, and we're going to 4 lots of 23. There's a video here to watch as well. Now, the similarities between the 2 is that they're both involved in the production of new cells, and they both start with 1 single parent cell. They had very similar stages to them, and the processes were the same with the same machinery. So this was a very long and detailed lesson. It's important that you go over this and have a look at the different videos and representations of this. Remember that the goals of what you need to know from this is you need to understand the role of homologous chromosomes. You need to describe the processes of crossing over and recombination and demonstrate how that contributes to genetic variation. Without understanding mitosis and meiosis, you really can't do those things effectively. There are some review questions in your Pearson book on this, and I will upload the handouts for you to work on.